presentations. Henrique and Arthur, I will introduce them a little later. I thought I'd start out with a few general comments. Both of them arrived at Iowa State in 2015 with Brazilian fellowships, and that was at a time when Brazil was a little bit more flush on fellowships, I think. Today it's a little bit more difficult. So I was lucky that they decided to join the group and join research around double haploids. Double haploids is a topic that has some history here at Iowa State, but no tradition. So in the 1940s, there was Sherrod Chase, who did pioneering work around haploids and double haploids in maize. However, it wasn't really continued. When he left, it was gone. And for some reasons, we started establishing a double haploid facility, collaboration with Kenny Gardner, the GEM project. And at that time point, Sherrod Chase was figuring out there's something going on around double haploids at Iowa State, and contacted me at the time, and then came here. And the students also had the opportunity in 2017 to meet with Sherrod Chase, and I think that was a really exciting event, kind of living history, if you want.
Yeah.
Arthur and Henrique are kind of the second generation of PhD students who work on the double haploid topic. The first generation was Gerald de la Fuente, and he went and left with open questions, as it usually is, which were picked up by Henrique or Arthur. And with this, I want to start introducing Henrique. He's coming from the very south of Brazil, I think the southernmost city. And he also got his undergraduate degree in Brazil, and he will explain it in more detail soon. Henrique has been involved in our inducer development activities, so developing inducers that are better adapted to Iowa conditions, that have a high induction rate. He also was looking into heterosis, if that could be used, inducer-donor interactions, and started with a GWAS panel, which will then lead over to the third generation of students. There will be some overlap with somebody coming in this summer, and he will talk about that in more detail. And I asked him to provide a few more details about himself.
Right. So, good afternoon, everyone. Thank you all for coming. Thanks, Thomas, for the nice introduction. Actually, I'm from the southernmost state of Brazil, but not the southernmost city. Well, even though it's close to Uruguay. And yeah, I did my undergrad in Brazil, did a short, exchange program at Illinois State University during my undergrad. Then I got used to the American system and things like that, and kind of thought it would be a good idea to do a PhD here. And gladly, Thomas accepted me, and everything worked out. Well, I'll jump ahead into my presentation. Today, I'm just gonna focus in one of my research projects. Thomas briefly mentioned all of them, but the one I would like to talk with you about today is a project designed to identify the genetic basis of maternal haploid induction. So, as you probably know, the DH technology is widely used in maize, but also in other species such as barley and canola. And a lot of research has been done into develop an efficient DH system in species such as rice, sorghum, oat, rye, and sunflower. Although the mechanisms in which haploid induction is different in each of these species, in maize, haploids are derived from crosses with maternal haploid inducers. And also in maize, haploid induction is caused by a 4-base pair insertion in a gene that codes for a pollen-specific phospholipase, which was discovered by a researcher from Syngenta in 2017, and it was named matrilineal. The insertion on this gene causes a frame sheet mutation that leads to an early stop codon. And that's how haploids are created in maize. However, even though that gene is necessary to produce haploids in a reasonably high frequency, the frequency of haploid seed over the total number of seed producer following a cross-pollination with haploid inducers is known to be controlled by multiple genes. And this frequency is commonly referred to as haploid induction rate. And even though now we know the gene that causes haploid induction, we still don't know how this gene performs. And there are two theories to try to explain how haploid induction occurs. And one of them is the theory of chromosome elimination, that the inducer chromosomes get kicked out of the zygotic cells during their development, and this theory was already proven to be true. However, the fact that this theory is true, it doesn't exclude the possibility that other processes also led to haploid formation. And another theory to explain haploid formation is the theory of single fertilization. And as you know, in corn, double fertilization is the normal, where one of the sperm cells fertilizes the partner nuclei and the other sperm cell fertilizes the egg. However, there are being reports that some of the sperm cells of the inducers are defective, and because of that, they may fail to fertilize the egg cell, leading to the egg cell to develop parthenogenetically, and this creating a seed with a haploid embryo. But before I continue talking about haploid inducers, I just want to show a brief overview of how the DH technology works in maize, for those of you that don't know. So this technology generally starts by creating new genetic variability, such as by crossing elite lines in a breeding program, and then the progeny, either the F1 or F2, is usually pollinated by a haploid inducer. And all inducers carry a dominant gene called R. Navarro, and this gene causes the pigmentation of both the aileron layer as well as the scutellum of the canals when pollinations go well. However, when the egg cell is failed to be fertilized or the inducer chromosomes get kicked out of the zygotic cells, the embryo becomes haploid. And this is how we identify haploid kernels. Since this is a dominant gene, it's very easy to detect contaminations, because if there is no pigmentation in the endosperm, they are not progeny of the inducer. So after haploid seeds are selected, they are usually planted in a greenhouse, and the seedlings are treated with colchicine, which is a chemical that acts on tubulin formation and leads to the duplication of the haploid of the cells. Our goal when we inject colchicine is to reach the floral meristems, because whenever the floral meristem that turns into a tassel is in the haploid stage, the plant will not shed pollen. However, if it is double, then the haploid plant will shed pollen. I will not talk very much about this step since Arthur is studying it, and we'll talk more about the limitations and opportunities in this part of the DH process. So after treatment, haploid plants are transplanted in the field, and those that shed pollen are self-pollinated to produce DH lines. However, the seed set in haploid plants is usually low, and normally one generation is required for for DH lines to have a reasonable number of seeds for testing. So now going back to the inducers, the first report of a maize haploid plant occurred on 1929 here in Iowa by two researchers. And since then, researchers from all over the world have been working on developing inducers with higher induction rate, and also incorporating phenotypic markers that allow the differentiation of haploid and hybrid plants in different stages of the plant development. one of these phenotypic markers is purple stalks. Other one that is very useful to discriminate seedlings before transplanting is the red root, that is also a dominant marker. And I would just like to mention some important achievements in inducer development before I continue talking about my research. A very important inducer was developed in 2013 by the University of Hohenheim in Germany. And these inducers has a very high oil content. And whenever this inducer is crossed with a donor parent that has a low oil content, that is common for the maize germplasm, it generates haploid and hybrid seeds with very distinct oil contents. And this oil content can be detected using equipment such as nuclear magnetic resonance. And because of that, this group in Hohenheim developed a machine that can accurately and automatically discriminate haploid and hybrid seeds. Also last year, a group from China developed an inducer that has two different fluorescent genes. And one of these genes has the expression limited only to the embryo. That is very good also for accuracy. And last, I will also like to mention about some of the inducers that were developed here at ISU by Yushi. And they are called the BHI series. And they carry most of these dominant genes that are using in the haploid discrimination. And they also carry the GA1 allele. And in case you're familiar with it, this allele is typically used in popcorn and sweet corn to prevent cross-compatibility with dent corn. However, if the inducer also have this allele, then the compatibility don't happen, and we can develop DH lines in these two kinds of maize. What is very interesting. But why is it important to develop inducers with a higher induction rate? Because it reduces the field space that is needed to produce the desirable number of haploid kernels, and also reduce the amount of time that is spent on detasseling, harvest, and haploid selection. I tried to make some calculations just to give you an idea of the cost to produce 8,000 DH lines with an inducer that has 5% induction rate and an inducer that has 10% induction rate. And just considering the cost for the field space and the time in haploid selection, considering that is done visually. So the cost to develop these 8,000 DH lines with an inducer with 5% induction rate is almost $10,000, whereas with an inducer with 10% induction rate is less than $5,000. This is more than is double the cost. So this money could be allocated for other projects of the bearing program, and how can I say, developing inducers with higher induction rate has a great economic impact. However, to effectively develop inducers with high induction rate, it would be useful to identify markers that influence the haploid induction rate of maternal inducers, and also assess the effect that this KTL have on induction rate in order to effectively incorporate them in some form of marker-assisted selection. Also, one of the objectives of my research is to identify promising inducers to be used as parents of new populations. And to do that, we tested a panel of 187 diverse inducers developed here at ISU and around the world. And all these inducers were crossed to a single donor genotype last summer. We used it as the donor an F1 hybrid called Viking 6001N, which was developed by a company from Minnesota called Albert Lee Seed Company. We had 2 planting dates of the inducer and donor, and in each of these planting dates, and the kind of The inducer of a given planting date was used to pollinate a specific donor planting date. And for each planting date, we performed 10 cross-pollinations. So haploid selection was done using the R-Navarro marker that I mentioned for you before that causes this phenotype. And for this project and some other projects I have, we selected more than 1 million kernels individually. So it was a lot of work. It required a lot of people and a lot of time. And after we selected the haploids, we had to verify the true ploidy of the kernels. And we did that by cutting the seeds and observing the embryo coloration. Whenever you have a haploid, the embryo is white, and whenever the seed has a hybrid embryo, it gets pigmented. However, unfortunately, the pigmentation of the embryo is not as good as this kind of. There are multiple genes that are into the anthocyanin pathway, and kind of variation in the number of these genes causes a difference in the pigmentation. So sometimes it's hard and tricky to verify the true ploidy of the kernels. And because of that, kind of all the verification of the haploid kernels was done by myself and by Vinicius, which is a visiting scholar from Brazil, and is seated back there in the room. And we used the average induction rate across planting dates as the phenotypic data for our GWAS analysis. And again, haploid induction rate is calculated as the number of haploids over the total number of kernels produced in a cross-pollination with a haploid inducer. And this number is multiplied by 100, so we can have it in a percentage. And the results of the preliminary analysis that I'm about to show you only include inducers that showed consistent data, haploid induction rate data across these two planting dates. And in this histogram, we have the distribution of the haploid induction rate data of all inducers tested last summer. It ranged from 0.6% up to 22.6%. And the majority of them are in this low end. And I believe that most of these inducers are some inducers that were generated on the background of 21 XPVPs that we didn't put, we did some marker-assisted selection, but didn't put too much effort on looking on their induction rate. And I also would like to talk about some of the inducers that are in the other extreme, the inducers with 20% induction rate. So if you remember, I showed you in the slide before that kind of currently the best inducers that are available are around 15%, but by crossing two inducers with a reasonably good induction rate, we were able to develop progeny that surpassed the 20% mark. One of the parents of these inducers was RWS, an inducer developed in Germany by the University of Hohenheim, and the other inducer was PHI3, developed by a private company from Romania, which unfortunately went bankrupt. And unfortunately, we didn't pay too much attention in other traits besides induction rate, and of the 8 progeny I tested last summer, just one of them still carried the red root marker. That is a very important marker for haploid discrimination. And here I have some pictures of the parents and also the progeny that was derived out of this cross. RWS has a slightly better induction rate than PHI3. However, it lacks most of the marker genes. If you see, the plant is totally green, as kind of a normal plant in the field. On the other hand, PHI3 has a slightly lower induction rate, but possesses most of these marker genes. In the progenies, we developed they carry all of these marker genes besides the red root. However, most of them were able to surpass the 20% mark. So for the genotypic part of my GWAS analysis, kind of SNP markers were generated using the DART-seq technology from CIMMYT. And with that technology, we were able to generate 51,000 markers. And after filtering for call rate and minor allele frequency, we end up with 6,500 markers. We probably will have to change that. That was quite a stringent filtering. And for further analysis, we probably are going to play a little bit more with that. Imputation and kinship was calculated in Tessel. Imputation used the method LD, K, N, and I. And kinship was made using the method center EBS. And the association analysis was run on GAP using the model farm CPU. And this Manhattan plot showed the results of my preliminary analysis. We found 3 SNPs that surpassed the FDR threshold, and they are located in different chromosomes. Unfortunately, this SNP we found in chromosome 1 is 20 megapairs away from the matrilineal. So it's possible that after, how can I say, I use my complete data set for the inducers, and after trying to correct for the misclassification rates, we are going to be able to reach the right spot in chromosome 1. But even though these were exciting results, after looking for genes in the vicinity of these markers, we found 5 genes that seem to present a phospholipase-related activity. That is the same activity of the gene, matrilineal, that causes haploid induction. So this was kind of a very exciting result, and we did some research to learn a little bit more about phospholipases. So phospholipases hydrolyze phospholipids into fatty acids and other lipophilic substances. And it was already reported that mutation on phospholipases delay the pollen germination and tube growth. So it seems that we are in the right direction. There are 4 classes of phospholipases based on the type of the reaction they catalyze. And one of these classes, called type C, it releases the acylglycerol and phosphate after cleavage. And the candidate gene we found in chromosomes 4 seems to have this type of function. So as general conclusions of this preliminary analysis, we found out that phospholipases may also influence haploid induction rate. However, further analyses are needed to confirm these candidate genes and the effects that they have on induction rate. And confirming these candidate genes is important in order to effectively implement marker-assisted selection in inducer breeding. And as I mentioned before, it's important to increase haploid induction rate because it has a great impact in the cost of DH line production. With that, I would like to thank all the members of the Luberset group, especially Vinicius, that has been helping me from haploid selection to verifying the ploidage of the seed until running the analysis. And Edi Carlos, who was a Brazilian and already returned to Brazil, but helped me a lot coordinating and performing the crosses in the fields. I would also like to thank the members of my POSC committee, Thomas, Danny Singh, Paul Scott, Somak Dura, and Bill Beavis, and my funding sources, such as the National Council for the Scientific and Technology Development of Brazil, the National Science Foundation, the DH Facility, and the RF Baking Center. And with that, I would also like to thank you guys for your attention, and I would be more than happy to take any questions.
Questions for Henrique?
Okay. Hi, Daniel.
Hello, thank you for your talk. Well done. I just have a really simple question.
Sure.
The single donor that you chose. Is there a particular reason why you picked that one?
Yes. So so the question was why we picked that donor, the Viking that belongs to a company in Minnesota. So we observed kind of a wide range of induction rates on that donor, and how can I say? It allows for this difference in induction rate to occur, because there is something that is called haploid induction rate that is caused by the inducer, genetics, but it's also there's something called inducibility that is due to the female genetics. So in case you used multiple donors or picked a donor that didn't allow such a big range, then our results wouldn't be that good. Hi, Danny.
Thank you. You made great progress, really exciting results. Two questions. You had F7 listed for progeny.
Right.
Is that the name you've given to that line, or is that a filial generation 7?
Yeah, filial generation 7.
Okay. The other question I have is, at what level of induction rate does it become detrimental? Is there a higher ceiling then?
Yes, kind of. I think we still didn't reach those levels, but I think that once you get to a level like 50%, it is going to become detrimental. And the reason for that is because the inducer, they're self-induced, and with that, for example, if you have a 50% induction rate and you plant a plot of 20 seeds of the inducer in the field, 10 of them are going to be haploids and are not going to shed pollen. So that compromises the ability to make crosses. Thank you. Hi, Ken.
Did I understand you to say when you looked through that series of derivatives from the Vikings, of the 8 that were taken forward, only one still carries the reference marker?
Yes.
Why do you think that is?
So kind of, I think the probably reason for that was that in the F3 generation, we advanced just 3 families. Kind of, we picked those that have had very high induction rate, and kind of we already noticed in the following generation that we barely, we almost lost the red root marker. That
Try worth evaluating for that trait as one.
so kind of. The F3 generation, was selected on 2015. That was the year that I arrived here. And at that time, I still didn't understand much about haploid inducers and things like that. Yeah. But that, taught me a valuable lesson. Kind of with that, I always try to keep some diversity when selecting lines.
Any other questions? All right, well, please join me in thanking